Hi everyone, my name is Shannon and I'm a PhD candidate working under the supervision of Dr. Bonnie Millard, and today I'll be showcasing some of the beef research being done in our lab. Producers want to raise healthier cows, both for animal welfare and because treatment costs continue to rise for the most prevalent diseases. However, animal health can be difficult to improve genetically because of low heritability of resistance to individual diseases, difficulty in on-farm recording, and the fact that selection for resistance to one disease may cause susceptibility to others. One easy way to solve these problems is to select for a trait that encompasses all diseases at once and is moderately heritable so that genetic improvement can be made. The high immune response technology is one such solution. It identifies animals with superior heritable immunity for both cell-mediated and antibody-mediated immune response, and allows for the calculation of an estimated breeding value for total immune response. This technology has been used in dairy cattle for some time. It was implemented by CMEX in 2012 for the purchasing of semen from bulls classified as high immune responders under the trade name Immunity Plus. There are many benefits of breeding for high immune response. There have been many studies performed in dairy showcasing these benefits, and I've highlighted some of them here. High immune responders exhibit decreased incidence of disease, strengthened response to vaccination, improved colostrum quality, and importantly, the reduction of the need for antibiotics, which resists the rise of antimicrobial resistant pathogens. Our goal is to translate the high immune response technology for use in beef cattle, and the first step to doing this was to perform a test on crossbred beef cattle at the Ontario Beef Research Centre, and this was part of a master's thesis of Nazrin Husseini. It was found that the high immune response method can be adapted for use in beef cattle, with comparable results to Holsteins, and with moderate heritability estimates that are similar to those found in dairy cattle. Another aspect of Nazrin's master's thesis was to determine the effect of heat stress on animals classified as high, average, and low immune responders. Due to the effect of climate change and the need for animals that are better able to adapt to heat stress during hot weather, rectal temperature was measured on crossbred beef cattle classified as high, average, and low immune responders using the high immune response test. Animals classified as high immune responders are better able to regulate their body temperature during times of heat stress, and this is an added benefit of breeding cattle for high immune response. Now that it has been established that the high immune response test works in beef cattle and is comparable to Holstein dairy cattle, the next step is to combine the high immune response technology and the currently available genomic selection infrastructure to develop a genomic test for immune response, and this is part of my PhD thesis. This project is in conjunction with CMEX, the Canadian and American Angus Associations, and Angus Genetics Incorporated. The goal is to immunophenotype around 4,000 purebred Angus cattle in the United States and Canada to build a base population for the genomic test. We will also be performing a genome-wide association study to identify key SNP markers and other pathways associated with enhanced immunity. We will be estimating phenotypic and genetic parameters for immune response, and also calculating associations with other important health parameters and disease information. This will be the first animal health or immunity trait included in Angus genetic evaluations in North America, with the goal of decreasing disease incidence and improvement of animal health without the increased use of antibiotics. Preliminary heritabilities for antibody and cell-mediated immune response have been calculated for approximately 2,000 cattle in Canada and the United States. These results are promising and indicate that antibody-mediated and cell-mediated immune response are moderately heritable and have the potential to be included in Angus breeding programs, with the capability of increasing animal health and welfare, producer profitability, and the sustainability of the industry. Thank you very much for listening, and if you have any further questions, feel free to contact me at my email, which is on the slide here.